Okay, thank you very much. And it's always been a pleasure for me to go to these biophysics scale meetings in Whistler, and we had the one in Israel, and it's sort of a home base for me for the kinds of things that I like to do. So my topic is a little broad, and we'll narrow it down momentarily, go from physics to physiology. On the left is a protein sequence of a protein called Bacterial Rhodopsin that I worked on for my PhD thesis when I was at Berkeley with my thesis advisor, Bob Glazer, who Mimi knows well. And on the right is more recent results that I've got working with some fellow scientists, Daniel Wankersut and Michael Kuhl from Denmark, to actually define three-dimensional coral polyps. And we'll learn more about that in a second. So, okay. Advance. I guess this. Okay, so this is purely conjectural, and you may argue with it, but I think the broad terms are actually factual. And the question about oceanography that was sort of that when I first came to Scripps, I'm not really trained as an oceanographer. I started talking to a bunch of biologists, and they were saying, well, there's sort of three questions. What is there? How much of it is there, and what is it doing? And so how much of that do we know? Well, I think as we do more eDNA, we find more creatures that we didn't know. So depending upon the broadness of your categorical sort of inference, you can think that there's a lot that we didn't know, or we know most of what's there. I wouldn't argue either of those points. How much of it? I think abundance estimates have been hard. Many creatures run away. There's acoustics, there's optics. Again, maybe quantitative PCR will allow us to figure out more about what is there if we can get rate coefficients for dispersion and also for dissolution. But for me, the really interesting thing is what's it doing? And so I just wanted to emphasize that the ecological mantra is really monitoring mass and rate coefficients through ecosystems. And I would claim that we really don't know as well. We certainly don't know as much as we would like to. I can't claim to have a generic understanding of every program around, but for the most part, I think we're lagging. The copepods are there, then they get eaten. The phytoplankton are there, then it goes away. And hopefully some of the new NASA satellites and other processes will help us to understand what's going on. So that's just sort of my intro. And I guess I just wanted to emphasize that my career has been a bit, I wouldn't call it strange, but I really like inventing instrumentation. And I've always tried to, and been successful, I would state, in collaborating with scientists. And so part of my heroes, if I go back to medieval times, was Tycho Brahe, this crazy Danish nobleman who ran around collecting extremely accurate information about trajectories of planets that were then used by Kepler to infer the elliptical orbits that were then used by Newton to get the laws of gravity. And then, of course, the latest thing was this amazing James Webb telescope. So I thought, first time I went out on a cruise, there were a lot of people, and scientists were pretty much inventing their own instruments to study what they were seeing, and the engineers were not as actively involved as perhaps I would have liked them to be. So that's been my sort of career goal, is to invent new instruments. So back then, in the mid-70s, when I went to Berkeley as a biophysics student, perhaps I'm the only one in this group that actually has a biophysics degree, but that's more of a formality than anything else. There are these hypersaline ponds, which to satellite look like purple or red, and they're actually a bacteria that can tolerate this really, really high salt concentrations. And it's a bacterial BR purple membrane. And so when I was in graduate school at Cal, this was really the first high resolution membrane structure that was being deciphered. And when we think about high resolution, we're not talking about light, we're talking about electrons. And as you know, the wavelength of electrons can be very short. So we can actually discern the alpha helices and beta sheets that are important components of protein structures. So the link to cryo-EM was through my advisor, Bob Glaser, who was very interested in understanding protein structures. And it turned out that the bacteria, that the purple membrane, there was a membrane-bound protein called bacterial rhodopsin, much like the rhodopsins in our eyes. So if you're familiar with that, it's basically a Schiff base that has an isomerization, and it does this amazing thing of taking a photon and taking that absorbed energy and transferring a proton or some fraction of protons across the membrane that then drives the energy of the cell. So it can form two-dimensional crystals. And why that's important, if you bombard it with electrons, you basically blow the thing up. So the idea is you have a repeating structure, and then you can actually use low doses of electrons and do what's called diffraction to see the crystalline pattern of that. And that gives you the amplitude.
I just sent me the link to a talk
Of the Fourier transform. And then if you can actually get the images, then you can get the phases and figure out what the structures were. So Bob was really interested in cryo-electron microscopy. We were in the Lawrence Berkeley Lab 1, which was where E.O. Lawrence had put his first cyclotron, and we were developing cryo-electronic methods because Bob said, look, if I freeze this stuff, it'll probably survive the radiation damage longer. But how do you prepare a molecule that lives inside a membrane for a thin layer that you can then see using electron microscopy, or electron crystallography would probably be the right word. So you do crystallography of two-dimensional specimens. Here's kind of a funny picture of me before Jules. That's actually me. And here's after Jules. That was me a few years ago. I take the liberty of showing a picture of me a few years ago. And what people were doing, Richard Henderson, a very famous crystallographer at MRC in Cambridge, where Perutz invented all of the crazy stuff that allowed us to see protein crystallography structures from three-dimensional crystals. They were preparing it in glucose. And Bob said, well, maybe we could prepare it in vitrified ice. And at the same time, I was writing a whole bunch of computer programs. And I was actually successful in preparing, this is the membrane. These are the alpha helices. There are 7 of them that go through the membrane. And these are the amino acid chain that connects this all up. And then I was able to figure out a way to prepare it in vitrified ice. And then what I was able to do was subtract the three-dimensional structures. So here we have it in glucose, which is gooey stuff on the outside of the membrane. And then we have ice, which has a lower density, non-invasive estimation of coral polyp volume and surface area. And this was published this past year in Frontiers of Marine Science. So what did we do, and why is it important? So surface area is super important for various types of organisms, especially for corals, because as you know, the corals live in the saline environment and they have to transport. Okay, so I think the key is don't touch your computer now. You promise me you won't touch your computer?
We promise you, yes.
Okay. Thank you. All right. Hands off. All right. So importance of surface area, especially for corals, transport and diffusion of gases across their physical boundaries, rates of photosynthesis and respiration are, as well as photosymbiont density and pigment content, are generally normalized to coral surface area. And estimates of coral surface area are largely rely on indirect measures of coral skeleton. So if you're familiar with the way this business has gone on, when I was the editor-in-chief of Oceanography, we had a number of papers on estimating coral surface area. And for the most part, people would use a wax dipping method, which was applicable to the coral skeletons. And then you could somehow decipher what actually the surface area was. But of course, you have an organism that has a lot of soft tissue. And when you look at a coral polyp, you imagine that those little tentacles, the little fingers that are sticking out, can actually, can you turn your audio off, please? That those coral surface areas, the actual fingers that are sticking out, actually have a lot of surface area. And so the estimate of soft tissue coral surface and volume was something that needed to be done. And when I was talking to my colleagues, Daniel Wankersud here at Scripps Oceanography, we had a technique which would perhaps allow us to measure the coral surface area. So they use something called optical coherence tomography. And I think many of us in instrumentation believe that there's a lot of stuff going on in the medical field that we could use in oceanography. And here's an example of that. So Daniel Wankersud and Michael Kuhl basically said, wow, this is really interesting. Perhaps we could use the same technology that's used at the time to look at the people's retina. I actually had my retina looked at last summer. Thankfully, that was still attached. There were no problems. And basically, it's sort of a LiDAR, if you're familiar with the pulse of light, that can be scanned in 2 dimensions. And if you could get temporal resolution, you could tell how far away stuff was. So imagine a little scanning thing that was able to then you create a 3-dimensional matrix. So an array X, Y, Z, and you scan in X and Y, and then the light comes back as a function of Z. So you actually get backscatter as a function of range in the infrared, except of course the beam is attenuated as it goes through, because of the backscatter, like layers that are kind of on the top are reflecting light more. And then as the beam goes through, it's actually attenuated as well. So many summers ago now, more than I'd like to imagine have passed. Well, this is a nice picture from Daniel Wankersud and a number of other collaborators, including Michael Kuhl, who many of you may know from Denmark, who've both been important coral researchers, where they basically used an OCT machine and said, wow, this is really cool. We can see the soft tissue surface. So we said, okay, let's borrow Michael's machine in California. It's a desktop machine now. You don't have to put the coral inside of the goggles because it's used for other things. And we used it on a coral called Stylophora. And this is just a picture that we took maybe 10 years ago from an underwater microscope. This was Tali Tribitz and Andy Mullen. And just to give you orientation on what we're looking at, and this is a wonderful picture, I believe, is on the cover of a few magazines. You can really see the symbionts, these little 10-micron dinoflagellates that live inside of the coral. And also this green fluorescent protein, which has become an important way to assay rate processes in cells, and with an award to a Nobel laureate who was a former UCSD scientist as well. Okay, so Stylophora. So we got this specimen of Stylophora, and what you're seeing now is I took the 3-dimensional matrix that came out of it and projected it in the Z direction, and the thicker parts are more yellow, the thinner parts are deeper blue, and by hand, I'm not embarrassed to go in and sort of see if I can do things that would have taken me years by the computer by just outlining. You see here there are 17 polyps. And what you see, interestingly, is the sizes. There are different sizes. Some are further away, some are closer, some are tilted. and I isolated these middle ones, 5, 6, 7, and 8 for further processing. So that's what I want to show you. So here's a height map of this coral. And it was really fun because we could really start to see the 3-dimensional structure of the polyps coming out by just doing something simple, by detecting a difference in the Z direction. I don't want to go into too much of the image processing because we're really more about the biophysics rather than the computer science. A lot of it was really trial and error, but it consisted of matrix segmentation, region filling, edge extraction, and point cloud differentiation. And here I'm just showing you. So now you have the polyp. Imagine a polyp, and I'm just going to take a slice through that polyp, a vertical slice, showing you the tentacles here through this polyp, and then using the physics, okay, physics to physiology. This is the integrated value along that slice in the Z direction. That allowed me to compute a interesting quantity, the total light incident as a function of XY on that Z stalk. And then that allowed me to then renormalize the coral and then start to fill in areas of the polyp. And then further processing allowed the definition of the inside here, but not the bottom inside. And then some edge extraction and then filling things in. And finally, the final result here is the actual outline of the shape of the polyp. And I hand labeled what I thought was the bottom. And it depends what you think of bottom. So these things there's a bottom and under the coral, as the coral ecologist had told me. But this was just kind of where the polyps ended. So that's all about the image processing. It wasn't done overnight. It was a rather laborious process. Finally, it allowed me to create these pictures of the polyps themselves. And so what you're seeing here is a single polyp. I think this was polyp 6. I got to know it well. And what you're seeing is a point cloud. So one of the processes in doing this work was really for me to learn about point clouds and also to educate my collaborators about point cloud. So a point cloud is just a point in 3 dimensions, X, Y, Z, with a value. And the value can be a color or it can be a scalar. And here what you're seeing is I encoded the amount of backscatter that I could see from that point in a common MATLAB map. If you're familiar with MATLAB, this is the hot map. And what you're seeing is that the tips of the polyps, the tentacles that were actually perpendicular to the incident beam had a higher backscatter, but we haven't learned anything about the polyp yet. And this is kind of a fun movie. I know movies don't play well in Zoom, so I'm going to just start this and stop it. So I hope that's going okay for you. There actually is a new app that allows people in Zoom to show movies, and we've tried it and it works pretty well. But it was really fun for me because I've done a lot of work in communicating science to the public. And for me, the 3-dimensional visualizations, we've invented these stereo microscopes for zooplankton and put them in the aquarium here. So what did we learn? Here are the 4 polyps, 5, 6, 7, and 8. Well, one of the important things was to do calibration. So we actually put in borate spheres, little teeny spheres of known diameters. We put fish eggs in, which were kind of squishy, and they squished down and created these hemiellipses. And what we tried and were successful in doing was using analytic formulas for hemiellipses and comparing that to a triangulation here. So Delaunay, I apologize to the French colleagues or Swiss French-speaking participants in this call, was a very famous mathematician who figured out how to tile surfaces. And we used that to create the surface area estimate. And then that was actually compared to an analytic estimate. Since they were squishy, we couldn't use spheres, so we approximated them as a hemi-elliptical. It's a fancy word for just saying a squished sphere. And we got pretty good results for the surface areas that were computed using the MATLAB programs that allowed us to define the surface area with a simple formula that allowed us to compute the surface area of the borate spheres. They were not squishy. We got better results on these. So we thought it was working pretty well. We were convinced that it worked well. So now, for the final slide of this section, what we're showing here is the 4 polyps and the surface area here. And there's a bottom that had to be subtracted. But what I thought was more interesting was the ratio of surface area to volume. And of course, that they're not the same dimension. Surface area is in, let's say, microns square, and volume is in microns cube. So that the dimension of that would be 1 over microns, or 1 over the metric. And interestingly enough, and I'm not claiming this would be a ubiquitous result for all of corals, but the ratio was pretty constant. So, and it's a small number. So square root of n is 2 if you have n equal 4. But I still think it was interesting, and the methodology, I think, is interesting as well. Okay, so the final part of this talk is a new microscope that is under development. And here's Orban Zvi, our wonderful postdoc from Israel, who has worked on corals for many, many years with our colleagues and friends in Israel, Yossi Loya in particular. And so now we're going to physiology. And you're probably aware that there are two techniques for looking at photosynthetic efficiency. One is the pulse amplitude modulated technologies, which were developed in Germany, and the fast repetition rate fluorometry, which was developed, I believe, by Falkowski and colleagues at Rutgers. And there are a number of systems that actually do pulse amplitude modulated work. There's an imaging PAM. So what we're doing is we're actually looking at photosynthetic efficiency in small images. And then there's another PAM that perhaps you're familiar with, we use underwater by the same German company, which gives us a macroscopic integrated value of photosynthetic efficiency. We thought, well, let's take the microscope that people are using in the lab and get it to work in the ocean. And we're close, but I did say we're doing preliminary results. So here's a single coral polyp, Stylophora, if I can get the Greek words correct. And what you're seeing these are the little teeny symbionts. These are 10 micron fluorescent dinoflagellates, apparently lost their flagella. And this is one single picture from a focal series. So here's the microscope, not a very exciting picture, but it gives you an idea what we're doing. You'll see this is in inches. I apologize. We can multiply by 2.54 to get centimeters. It's small enough for a diver to carry down, and it builds on previous work that we've done with a benthic underwater microscope that we published some years ago. And my interest has been in underwater microscopy, bringing the microscope into the ocean. Here's another picture which shows sort of how it works and the important ingredients. We have a ring of LEDs here that are providing a strong pulse of light. And we have these linear actuators. So when you have high magnification, you have very short depth of field. This is a function of the physics and the laws that govern imaging. And then, so we want to be able to get a focal plane series of what we're looking at. And then a bunch of batteries, and here it is in the lab. Not a very nice picture, but actually this is in the lab and it's being tested, and it's pretty much working. So we have colleagues at Scripps, Jen Smith, who's been working with the macroalgae, and or was up working in their lab and found a gametophyte and a sporophyte from a baby kelp, I say the baby macroalgae. And now these are the first images of the photosynthetic efficiency that we've been getting from these, and we actually measured it over a 2-day period, and we could see the decrease in photosynthetic efficiency because it was just sitting in a fish tank in the lab. So we think that's working well now, and we're struggling a bit with transforming this. It's a much more complicated scheme because now you're using, pulses of light and saturating pulses, and then you have to look at you saturate the photosystem and then you hit it again, and then it dumps the energy into fluorescence. So it's a complicated procedure, which we are getting a handle on, and we think basically we're getting it to work. So hopefully in the not too distant future, we'll have actually individual zooxanthellae from the real world. And so I've been having a lot of fun. This is now a single plane in a 3-dimensional matrix using my MATLAB expertise, if I can call it that, to extract the locations of single zooxanthellae. And this is now a composite from the entire volume. So each slice is a different focal plane. And if I process all of those focal planes, I can extract all of the 3-dimensional locations of the zooxanthellae that are in focus. And my plan is to make these into a 3-dimensional picture. I was hoping to have it for today, but I was even happy to have just this simple result. So what might we, current diving PAMs integrate the estimate of photosynthetic efficiency over large numbers of the symbionts. With our new benthic underwater microscope for pulse amplitude modulated imaging, we hope to get volumetric abundance of the symbiontinia, 3-dimensional locations, characterization of the heterogeneous surface structure that leads to photosynthetic response, and likelihood of bleaching, and characterization of photosynthetic efficiency. I mean, I think simplistically in my mind is the coral kicking them out when they bleach, or are they deciding to leave on their own? And maybe that's an oversimplistic characterization of the process. So I'm happy to entertain any questions now. And I would say that one of my interests has been imagining what it would be to be a micronaut. And so we actually built a game, not finished, but these are the three-dimensional corals. And what you're seeing here are these little symbionts kind of floating around in 3 dimensions. So I would be happy to answer any questions, but I think it would really be fun to get into gaming in a way that, you know, when you learn the rules of the game, you're actually learning science. So happy to take any questions now. Please go ahead if you have. Okay.
Roberto, are you back? Okay, thank you so much for this amazing talk, Jules, and apologies for the technical difficulties.
Okay.
Roberto's laptop seems to be crashing today. Sorry about that.
All right.
Are there any questions from the audience? You can just unmute yourself and ask the question. I will start. So with your last point about the symbiotes either being kicked out by the corals or leaving by themselves, how do you imagine actually analyzing this type of data? Would this be like 3-dimensional trajectories of symbiotes leaving the coral?
Well, I guess no. I guess my thinking is something like if the photosynthetic efficiency is maintained in the presence of bleaching, then it seems like the symbionts are still pretty healthy, but they're being kicked out by the coral. But if we see a drastic reduction in symbiotic in photosynthetic efficiency, we can imagine that there, you know, I'm not an expert in coral physiology. There are many people perhaps on this Zoom which would tell me more, but that's a sort of interesting idea. We do have from previous 3-dimensional recordings. I'd be happy to share them at a future date with anyone interested. Pictures of zooxanthellae aggregating subsurface and then being expelled through the polyps. So yes, the possibility for tracking them, if we can get the frame rate up, is a sort of enchanting, I would say.
And the size is 10 micros, 10 micrometers?
Yeah.
Okay.
Yeah, pretty much. Yeah.
Well, that's amazing.
Well, the diffraction laws hold. It's just a matter of getting close. If the physics says you could do it, and if the funding is there and you have enough smart people that are not me, I'm not the optical engineer, but Paul Roberts was in my lab, and now we have a few others. Joe Snyder, who's been helping, and or is our resident biologist. So yeah, I mean, that's the sort of, I'll use the word sexy part of this, is to see stuff underwater, rate processes that, and doing it in situ at the time of bleaching, I think is important. So when we built our benthic underwater microscope, we actually went to Hawaii with Jen Smith, one of our coral researchers, and we were able to characterize the algal overgrowth of the corals as a function of space as they were bleaching, as a proxy for the temporal changes that occur. So we traded space for time and got some nice pictures of where actually the algae were settling on the corals. And that's in our NatureCon article from, I think it was 2016, if folks are interested in going back to that.
And what you mentioned the volumetric estimates of the algae. Do you have any? Have you quantified this already, or this is yet to come?
Well, over the weekend, I was finally able to identify the locations of the symbionts, and I now have a 3-dimensional matrix. So that's what I think is fun, and that's next on my agenda.
Okay, so there's more than in the game.
Yeah, okay. Yeah. For sure. Okay, anybody else? I'm happy to hang out. Any questions? Any more questions? And
more questions? I don't think so.
Okay, so I guess you would ask for a half-hour talk. I think I did keep it to pretty much of a half hour, and
yes, wonderful. I guess we can then stop it here.
okay.
Thank you so much, Jules, for this beautiful talk.
Okay,
And
more than
we'll
welcome.
follow up with the question about the tracking bacteria in situ.
Okay, my pleasure to collaborate with you guys if there's interest in that.
Thank you very much.
Okay. Have a good night to those of you in Europe, and good morning to the rest of us, or midday to the East Coasters in the United States.
Okay.
See you guys later. Thanks for the invitation. I very much appreciate it.
Thank you. Thank you again. Have
Okay.
a good day, everybody.
Thank you.
Thanks, Mimi. Great to see you.
Good to see you.